Okay, everyone, I think we'll
Mhm.
get started. I think we have a fair number of attendees already, so we'll just get started. So, hello, everyone. Welcome to CelVivo's inaugural seminar series, where we will be hosting a talk once a month focusing on topics relevant to 3D cell culture. Thank you, everyone, for joining us from different parts of the world. We're so thankful to see you all here today. For this first installment of our webinar series, we have Professor Krzysztof Wrzesinski exploring the scientific advantages of 3D culture and the ideal culture conditions. So before I introduce our speaker, I wanted to ask all the participants to please submit if you have any questions into the Q&A function located at the bottom, and we will address them at the end of the talk. So to get started, I'll provide a brief introduction for Krzysztof. So Krzysztof began his academic journey at the University of Southern Denmark, where he pursued the establishment of physiologically relevant human in vitro models for preclinical drug development. Since then, he has been immersed in biomedical science research, focusing on advanced cell culture technologies and development of robust drug testing pipelines, employing different in vitro models. He is the co-founder and chief scientific officer of CelVivo and the extraordinary professor at the Northwest University of South Africa. Please welcome Krzysztof.
Yeah, thank you very much, Rika, for the very nice introduction. And I would say good day to everyone, and thank you for using your time to be here today. In today's webinar, I would like to present you my thoughts about why we actually change from 2D to 3D. And let's start with this, that in our laboratories, we create models to mimic in vivo organism behavior. Most often it is actually human body we are trying to mimic. And to be specific, quite often we are not going for the whole body, but we are trying to mimic and part of it. So organ or tissue is our actually modeling target. And even so, even such a simplified model are very complicated. And as you can see on this drawing of the liver tissue, in vitro model will need to recapitulate several functionalities. Just to take the most basic ones, if we think here, the model will need to recapitulate how we deliver the nutrients, how we deliver necessary gases, and how we deal with the secretum of this tissue, in this case, bile production, for example. And if we think about adding different cell types, this adds another level of complexity. From a historical perspective, we used to use monolayer culture, so-called 2D cell culture, to build our models. Those are fairly easy to operate and use very little complexity systems. And that actually can be an advantage for us, especially during the data analysis, data coming out from our experiments. However, the lack of the complexity is also a limiting factor for those types of models. Cells, to function normally, accept all the nutrients and gas exchange. They also need a communication. And in traditional 2D, the communication is a very, very limited factor. And additionally, due to the technological necessity, we actually regularly destroy those communications. And I would say that more about this you can easily find in one of our articles in toxicological research. You can see the link or the reference here. And as you can see, this is a quite historical publication from 2013. So going to the 3D models, meaning creation of the biological constructs with a co-localization of cells in the three-dimensional structure. This creates a physiological environment which promotes cellular communication and develops a tissue-like functionality. Again, much more we have to describe about this in this rather long time ago published article. But we actually postulated that allowing a extensive cell-to-cell communication and controlling how we deliver nutrients to cells, we can actually trigger so-called metabolic reprogramming, or otherwise named physiological functionalization of our in vitro models. Beside proper nutrients delivery and spatial localization of our cells, the adequate time is actually needed to develop proper physiological relevant models with right cellular communication and functionality. Unfortunately, many tools we have been using had several limitations in this aspect. And those limitations actually trigger us to develop a new methods which improve our models and place them closer to a physiological in vitro situation. Going a bit back to standard 2D based methods, we all know this type of growth curve with a lag, log, plateau, and decline phase. We, from physiological point of view, for most experiments, the plateau phase would be the most preferable to be used. Unfortunately, due to technological aspects such as controlling cellular population or proximity of the decline phase, most of our 2D-based experiments have been shifted to a log phase, where from the technological point of view is the easiest to control phase in our 2D in vitro systems. Constructing the 3D based models allow us to actually create prolonged plateau window, and we also are able to control it better. In a case of our liver model, we have documented stable plateau phase for over 21 days of the functional equilibrium, we so call it, or in given a 42 days of culture period. This stable reactive window allow us to perform physiologically relevant repetitive experiments. That was very important for our research. And then coming back to the 3D cell culture and tissue models, we can divide the models based on the origin of the material used to create the constructs. So we have our spheroids, and normally or often they would be created from immortalized cell lines. Then we have an organoids, which will be made out of different types of stem cells. And we also have this 3D primary cell culture derived directly from patient of animal tissue. We also will hear a term of tumoroids, derived from tumors, as well as, for example, terms are like an assembloids, which basically are constructs made by fusion of different organoids. And yes, this is just a very general classification, and I know, and you also know, that many different variants and shades of this classification exist in scientific literature. But it's just to get some kind of feeling how what we have, what is available. But we can also create a different division, another type of division, and this one is based on the technology we use to support our living construct. And in this division, there are basically two main groups. One is called static culture, where nutrients exchange is based on the passive diffusion, and the other one, which is called dynamic 3D cell culture, where the diffusion is improved by introducing technological functions such as agitation or media flow. Despite the method we use to create our 3D models, we have to think about basic two things, two goals. It's a structure formation and nutrient supply. Yes, we would like to build a structure because we would like to have our cells in a real 3D. And yes, we need to think about delivering a right amount of nutrients and gases for a right amount of time to be able to allow cells to create complex cellular communication and functionality. So our starting material, our sample, can come from various sources, but they are most often a single cell suspension after we finish the processing. So after we finishing digestion of the organ or preparing a biopsies. We need to give them time to recreate or establish this cell-to-cell interaction in our new systems. And often we are using a different type of support systems. So we can use scaffolds or hydrogels to help to create this extracellular-like environment where cells can grow into the 3D structures. In some cases, we also use simple ultra-low adherent plates or micro pattern plates to create or to help or to force the cells to create a conglomerates. Actually, use of the plates is a relatively simple method, and it actually gives to the cells a so-called peace and quiet. And so they have time to establish the structure. However, the problem in that approach is often that if used in a prolonged time, we are often coming to a problem with delivering enough nutrients and removing the products. So because we are using a passive diffusion, and often we also have to deal with the high capillary forces of the small UL plate or micro pattern cavities. So when we have this passive diffusion, we are able to, the cells are able to access only nutrients from a direct neighborhood. And within the time, they will create so-called nutrient depletion zone. And this zone will be replenished by a movement of components in media. But because this movement is only a passive, that will take some time. It will basically create this depletion zonation. This is actually a limiting factor for creating models using the static system. And this also limits the physiological relevance of such models. All right, so we would like to kind of overcome this hurdle, and we do this by introducing a media movement. So by introducing the media movement, we are removing the nutrient depletion zone, and we create better conditions for growth. However, introducing the media movement exposes the construct to the increased shear stress, and an excessive shear stress creates an unphysiological environment. So in principle, we have two situations where one offer a peace and quiet environment, but who has a low nutrient supply capacity. And then the second, where we have a very nice nutrient supply capacity, but we create turbulent environment with excessive non-physiological shear forces. And of course, we would like to combine the best part of those two systems. And I would say, yes, it is possible by using a clinostat technology. What is a clinostat? A clinostat is a device which uses rotation, a simple rotation, to distribute the effect of the gravity. The technology idea is actually quite old, as you can see. It was actually described first time in the beginning of 18th century by Denis Stoddard, and then it was made first actually working machine in late 19th century by Julius von Sachs. And then the whole concept has been kind of reinvented in late 80s by NASA to create machines which supports and space shuttle flights or experiments made on the space shuttle. So I would say we have definitely not invented the technology, but I would tend to say that we have mastered it. So what we do is that our clinostat system utilizes the clinostat principle, which allows to remove nutrients depletion zone from around the growing construct without actually increasing shear force. How we do it? The system basically utilizes a gentle rotation and gravity force to keep constructs in a suspended stage. The system utilizes closed cartridge principle with very minimal shear forces because we do not have a pressure difference. And here on this image, you can see how the constructs move inside the vessels, where when we remove electronically the rotation of the vessel itself. So CelVivo clinostat is a 3D cell culture system consisting specialized incubator and a drive system, and disposable culture vessel, clinoreactor. The principle of working with clinoreactor is a bit different than standard flask, but or cell culture flask, but I would say that it's almost as easy. This slide speed up video shows media change procedure in a clinostat, where basically we use a standard 10 milliliter syringe to remove and replace media, and then re-sterilize the whole system, and then that's ready to be reintroduced into the driving system. So this this procedure was a bit speed up, but actually a whole process usually takes them less than 1 minute. And here you have another video where we are actually showing how easy it is to actually sample or retrieve your construct from the clinoreactor. And the vessel can be very easily open and put into the so-called Petri dish mode, where you have full access to all cavity and you have no problem to removing any existing construct or sampling it or sampling media. Using the system, we are able to create uniform 3D constructs from individual cells and with very limited production of ECM. And this is actually a picture where we show, like, from single cell suspension through days of culture, we actually are able to create very uniform constructs with similar shape and size, which are useful for further experiments. You can also use different aggregation methods. Here is, for example, the picture we show how we can use actual micro pattern plates to create, some people say embryonic bodies, or we say it organoids or spheroid or construct seeds. We can also use the system with hydrogels, such as sodium alginate, for example, very popular, or commercially available vitrogels. You can find quite simple protocols how to actually utilize the hydrogels into clinostat systems on the CelVivo webpage or in our publications. And I would say that the hydrogels matrix are very useful when we would like to produce constructs made out of cells which basically completely do not like to stick together, or the ones which actually stick too much, and then you would like to control the number of them, and you would like to also control the matrix components. And here actually is It's a picture from one of our publications where we use a so-called two-component gel, Q-gels, to produce a very nice and uniform constructs. Uniform spheroids can be created from many different cell types. Here we actually have pictures which shows an population of spheroids created from immortal cell line, small cell, long cancer cell line. And what is interesting here is that all those populations are created from a different subclones of this one cell line, and those subclones are having a bit different characteristics if we think about metastatic potentials. And that also is reflected in the shape of the spheroids and easiness of the how easy you can make those spheroids. And here I also would like to show you another example where we actually created a construct made out of rat insulinoma cell line INS1E. And you can see that those constructs are not exactly identical or regular. So that presents a challenge with downstream applications if you have a population of not very uniform constructs. But actually, due to a clinostat principle or clinostat effect, by keeping those constructs, just basically keeping them longer in our system, you can actually achieve a very nice spherical constructs and very identical and still much more useful for downstream applications and for their reproducibility. So in clinoreactors, we create an environment with a so-called omnidirectional gravity stimulation. So what this means? This means that once the uplift due to the movement of the growth media match the downpull of a gravity, our 3D constructs will be kept in essentially shear stress-free suspension and will appear to be in the stationary orbit as here. Or if you will look on this electronically removed rotation of the vessel, they basically have a very little movement inside of the system, and those small movements allow to eliminate the diffusion depletion zone without increase or excessive increase of shear force. Using the system, we are able to trigger very pronounced changes in the cellular morphology. Here you have an image where we shown cells grown in the monolayer and on the standard plastic surface, or the same type of cells grown in three-dimensional structures, and the morphology changes are very pronounced. And we think that this is the key factor to determine why we start to see a tissue-like behavior in our constructs. So we have been able to demonstrate that constructs made out of this immortal cell line are able to regain a physiological relevant functionalities, and we are able to use them for a physiologically relevant experiments with a long-term treatment and repetitive treatment. And we also shown that they are actually predicting and known reactions of different drugs in a much better way than the same type of cells grown as monolayers. The clinostat or clinostat principle promotes those different cellular structure developments. And actually, here I can show you a short slide from research made by Agnieszka Rybak from Max Dulbrigor Center in Berlin, where they actually used the clinostat to create a construct without necessity of using an ECM-like gels, like a gel tracks in this case. So they have created constructs. It's an neuronal organoids, and they have actually compared the shaker and gel tracks with an clinostat system alone and only cells, and they compared several parameters, and they were very happy with the results. And they could say, okay, we can remove the artificial ECM-like component out of our model. And yet another point, it's like this is actually not so long published article from John Wu lab, and Carlos Pinson has been a person behind the experiment where they have created a synthetic embryo, or how they call it, a blastoid, out of stem cells made out of the bovine cells. And then very interesting, if you look on this picture, the synthetic blastoids are extremely identical with a native blastocyst. And that is something which was only possible due to use of this clinostatic effect in the clinostat system. So another researcher, where we have a researcher from Australia, Romal Stewart, and in this case, he actually produced a neuronal organoids, or some people will say mini brains, and he used them as a host for the virus infections. Actually, what he was studying a different infection rates of a COVID virus variant BA5 and BA1, and then they actually were been able to see that the variant BA5 has a much higher infection rate in the neuronal tissue and stays in the neuronal tissue for a longer time. Another COVID-related research is actually coming from Professor Doris Wilfangsteder lab from Medical University of Innsbruck, where they actually use a long organoid to study an virus infection, or COVID virus infection. And I would say about this one, I would very much invite you to our next webinar on November 15, when actually Professor Doris Wilfangsteder will be talking about her research in much more detail. So those were just a few examples how the clinostat system can be used. The clinostat technology can be used for many different cell types. It is easy to operate and adapt to already existing lab procedures, and it is able to produce reproducible and physiologically relevant data. And I think this is a very, very important thing, that use the in vivo functionality as your benchmark. So thank you very much for your attention, and please do not hesitate to contact us for our local distributors if you would like to have more information. And for those in America, I would say, please don't hesitate to contact Riham. She is actually in our US branch, in CelVivo US branch. She's our senior application specialist, and she has a very in-depth knowledge about the system and how you can use it. So once again, thank you very much for your attention.
Thank you very much, Krzysztof, for the talk. I know we're just at 9:30 right now, but we did have a few questions that came in through the Q&A function, so I'm going to quickly try to go through a few of those. I apologize if I don't get to your questions because we got quite a few. So the first question that came in, Krzysztof, is, can I grow patient biopsy samples, tissue samples, and primary cells in the clinostat?
So I would say grow, it's a very important word, because if we think about the tissue biopsies, we usually think about keeping them. So yes, we have examples where our users are keeping biopsies for extended time, and they are not only keeping them alive but also keep the functionality. And if you think about growing, so this is like an let's say we would like to have an outgrowth. Yes, that is also possible.
Great. Another question that came in is, which areas of non-clinical studies are being impacted by 3D models such as organoids in context of regulatory pharmacology and toxicology?
So I think this question refers to the FDA Novelization Act from I think last year, where they were saying that right now we are able to actually get an approval for new drug candidates without having need for the animal tests. And then I would say that's very nice, and I hope in future we really will go for this. For right now, I would say that we may minimize use of animals, but we will not be able to completely completely eliminate it, because our models are still too simple, but they are actually much more complex than the ones based on the monolayers. So if we think about a predictability, as I showed on one of our slides, that if you basically can go from from R square like 0.55 to R square 0.8085, that it gives you a much higher confidence, and you can actually eliminate a lot of unnecessary animal-based tests. So we are impacting this how well you are prepared to go to animal tests, to the preclinical systems, or from preclinical to the animal test.
Perfect. And another question is there a specific media that must be used to grow cells in the clinostar? And if I wanted to use a thickening agent such as cellulose, would I be able to add that to the media?
So again, two questions in one. First, no, you don't need a specific media to grow cells in the clinostar or clinostatic effect. That's the point, that we use a physics to keep cells in suspension, and then you can use all the media you are using in a standard, let's say, flask or petri dish-based systems. And yes, if you want to use a thickening agent, this is what I assume it's about, the cellulose fibers, they are often used as a thickening agent. Yes, you can do this. That's also possible. The clinoreactor is created that way that you can actually access it through several ports, or open it completely, and you can add whatever you would like to have it, whatever fits into the chamber.
Another question, how difficult is it to lyse these cells when performing molecular biology techniques such as PCR? How many cells are in one spheroid?
Yes, that's that's a nice question. So actually, we have published a protocol where we show how to do the enzymatic digestion of the 3D constructs, because the point is, like, first, are we talking about a native construct, so means the construct created only from the cells and the natural ECM you produce, or we are talking about a construct created with a hydrogel, for example? So if we talk about a native system, then a simple stepwise enzymatic digestion will allow you to actually create a single cell suspension, and even a viable single cell suspension. You can do a flow cytometry and on them afterwards. You can count them. So that's actually not a problem. I would say please look on the web page of CelVivo. There is the link to the publication. I don't know, Riham, because you probably see who has asked that. So you
Yes.
maybe can send the link to this person. And then
Yes.
the second part, if you use an hydrogels, quite often different type of the hydrogels, they have their own so-called releasing agent, where you can use it and get rid of your artificial ECM. But that will depend on which one you are using. And yes,
Another
it was how many? So that depends on the size. So the one which you have seen on the presentation made from HepG2s, C3As, those were around 80,000 cells if they are around 800 to 1 millimeter size. If you think about the primary hepatocytes, they were, I think, around 50,000.
Perfect. And another question that'll be of interest to you, how would one optimize the RPMs for my cell line?
So that's the whole thing about the CelVivo, and I would say I always that's not the first time I'm getting this question, and I always say, fortunately, the speed adjustment is based on physics, not on biology. And since we understand physics much better than we understand biology at current time, it's basically a function of mass, surface, and viscosity of your liquid. So once you rather establish it for the size and the density of the spheroid, not a type of the cells. So once you have this dummy made, then it will work. And again, we have on our webpage quite a few emails, movies, how you will basically see it. And currently, it's an operator who has to actually adjust the speed.
And another question from a familiar face, when changing the media, is it okay only to change out 5 mils rather than trying to change out as much as possible without losing the growing organoids? I need an answer based on your current experience.
So of course it's all right to exchange 5 milliliters instead of 10. What quite often why we change media? We change media because this is a cartridge-based system, and medium is our nutrient supplier and also a buffering for all the waste products our constructs excrete. Yes, so we need to exchange it when the nutrient capacity is diminished or the buffering capacity is diminished. And I know that quite often people actually don't change all medium because they would like to have some of those secretome left in the vessels, because this is actually the cellular signaling. This is this chemical cellular signaling as well, not only waste product. So of course it is possible, and I would say I would advise to actually make the kind of feeding growth curves or feeding capacity curves for media and see, okay, how I can do it. What we do with our media and our liver model is basically we check the glucose consumption and glycogen production. And then we create an feeding regime, because we also know that by changing the feeding regimes, you can actually change the behavior of the model. So we could have, for example, a lean liver or fatty liver made out from the same genetical background, just basically changing the feeding behavior. So depends if the cells go to the McDonald's or if they go to the vegetarian restaurant.
Thank you. Another question. Can you share some experimental and CFD simulation of shear force calculations in the clinoreactor?
Yes, we have a publication from some long time ago, and I would say right now we don't have the newest data. We are actually working on some simulations. So we can share some numerical data. Yes, that's what's in our publications. I think from 2018, where we did the calculations, and right now. You have to be a bit patient. Yes, so hopefully we will have some simulations, not so distant future, or animation of the simulations.
And another question. Do you use a syringe to change the media? The risk of scratching the plastic is a bit high, as I may have accidentally done it before. Are there any existing ori ducts to use instead?
So I don't know about organoids. Yes, we use a syringe, and syringe by itself do not scratch. It's a needle which scratch. So I will advise to look for an so-called blunt needles or a plastic needles. Quite often, medical Veflons, for example, from veterinary use, are a very nice substitute. Or you can actually try to feed long gel loading tips to your syringe, and then you will not be scratching the surface of the clinostat or clinoraptor.
Great. One other question. Can you present some protocol for patient-derived organoids using your platform without any matrigel?
So that's the example which probably is closest from the Agnieszka Rybak from Mark Stuhlberger. And I think we do have some applications on our webpage on that.
Perfect. We actually also have a comment in the Q&A from Hasitha, who has been using our product for quite some time. He said, blunt-ended needles works well to prevent scratching, and that's something that we can all attest to. Blunt needles work quite well.
Yes.
Yeah. And it's just a matter of practice to prevent the scratching, which I think Krzysztof can very well attest to.
Yes, and always, if you have a standard needle, if you cannot get a blunt needle, try to buy a bit thicker needles, and then the ones with this type of shape, and then you can kind of go gently to the wall. But we know that yes, the needle needs a bit of practice. So also the previous question about how much I have to change media. So maybe if you just do a normal maintenance, changing, let's say 80% of media is good enough. I know when you will start to do the experimental treatment, you have to be more precise what you do. But with the more normal maintenance, you can actually avoid this puncturing or scratching by changing a bit less.
Okay. Great. I think we have, one last question. What gauge size of needle is best in your experience?
That's a good question, and I think we usually say something like between 16 and 20. That's the gauge size. So if you will use very thin needles, you will have definitely less micro bubbles, but your time of exchange and your fingers will get tired. If you use a very thick needles with this low gauge, or yes, then they will basically introduce more bubbles, but you will be faster. So if you have a proper media, which is degas, does not contain a lot of micro bubbles, you can use a very thick needles, and it's easy to work with.
Another interesting question just came in. To what extent can one change the gas phase? That is to say, can one run hypoxic experiments?
Oh, so that's I don't see who has made the question. I don't have the view on that, but I would say I don't know how to answer this question officially, but I would say please keep an eye on CelVivo web site, especially in December, where this question will be answered in very much details.
Okay. I think that was the last question that came in. I know we're running a bit over time. We're about 15 minutes ahead of time. I mean, in extra time, not ahead of time. But if there aren't any more questions, then I think we will conclude our first webinar series for today. thank you all so much for participating. Thank you for being so active and, asking your questions. And I invite you all to join us again on the 15th of November for our second installment of this webinar series.
Yes, and if you have any other questions, please write to us, write to Riham or me, or
Yes.
just info.CelVivo or info@CelVivo.com, and we will try to provide you with the best answer we can have.
Yes,
So
and
thank you very much.
yeah, thank you so much. Bye.